Hello, my name is Theodore Alexandrov. I am a team leader at Amble Heidelberg, and today I will present you the progress of the Metabolomics use case in the European project Cloud Button. This presentation was prepared by me and Lachlan Stewart, who is a software developer in our team and the main team member involved in this project. First, let me introduce what is spatial metabolomics, because this is a technology which is key for our use case. And I'll explain you first what is metabolomics. Metabolomics in general is one of new omics fields of science and technology, which is focused on the study, analysis, detection of small molecules. This is an emerging field which has shown a lot of progress over the past years. And so it is recognized as a key to understand biology, to help in medicine, to discover drugs, as well as to develop novel therapies. And there is a lot of excitement and enthusiasm about metabolomics and how it can be helped to address challenges in cancer, metabolic diseases, as well as develop absolutely novel therapies like immune therapies. Spatial metabolomics is a subfield of metabolomics and is often considered to be one frontiers of metabolomics, where it is focused on spatial detection and interpretation of small molecules in their native spatial context. For example, in tissue sections or in organs, or in general, across different spatial scales. In some sense, one can think of spatial metabolomics as a way to provide molecular microscope, where we can see not only organization of cells within the tissues, but we can also see what kind of small molecules these cells contain, or what kind of small molecules they use to fulfill their functions. What is very important for the motivation of spatial metabolomics use case in Cloud Button is that spatial metabolomics technology generates big data. Since every pixel from a lot of imaging data generated by spatial metabolomics can be considered as a separate sample, with a number of pixels reaching as high as hundreds of thousands or even a million. So here you can see a recent review that we have published, which discusses spatial metabolomics, as well as imaging mass spectrometry in the current age of artificial intelligence. And in the next slide, I would like to present the technology, imaging mass spectrometry, which is currently considered and used as a key technology for spatial metabolomics. This technology is important to understand because our use case is focused on helping this technology and develop novel methods for this particular technology. So here you can see one of our team members placing a glass slide with a tissue section on top of this into an imaging mass spectrometer. Here you can see a visualization, an optical image of this section. This is a liver section. And then this instrument goes in a rasterized manner across the tissue section, and from every pixel, it shoots a laser at these pixels. The molecules get desorbed and they get analyzed in the mass spectrometer itself, generating a very long, almost 1 million values per every pixel spectrum, so-called mass spectrum, which represents relative abundances of molecules contained in this particular pixel. So after some spectral processing is performed, one can generate the images. And the key step in the whole pipeline is to really extract what kind of molecules are represented in this very, very complex data. And I'll not go into details what are specific complexities of finding these molecules, but I would like to stress out that this step, so-called metabolic identification, or finding again molecules in this data, is one of the key steps of making use of this data. Over the past years, we have developed several algorithms in particular the key algorithm published in 2017 in Nature Methods for metabolite identification in this field. And here I'm presenting you Metaspace, a cloud platform for spatial metabolomics, which can be used by freely by users across the world by taking the data, uploading this data to imaging and spectrometry data to our web app, where the processing is performed to perform this metabolite identification step to find the molecules and provide user results as list of molecules, images, and so on and so forth, but also store the results, in particular public results, into metabolite imaging knowledge base. In our team, several people are working on this, in particular software developers, including Lachlan Stewart, but also other software developers. And Metaspace as an engine and platform is funded by European Commission. It was created as a result of a European project, Metaspace, but it's also co-funded by National Institute of Health in the United States. And we are very happy and proud that Cloud Button has selected us as a use case, and we are working very closely, and we'll present the results of this project, how Metaspace can be performed in the serverless scenarios. But first, I would like to show that there is a lot of demand for the new capacity, in general for the use of Metaspace, but also for the new capacity. So here you see a screenshot from our web app. Here you can see the map of users. You see it's worldwide. There is a lot of users from Europe, but there are also users from the US, Australia, and many other countries. And here you see the number of data sets and the steady growth of the use of Metaspace across the past years. And currently, we have already performed processing of more than 8,000 submissions, which is a pretty substantial number for a relatively small and emerging field. What's important also, 50% of them public. That means they can be reused or reprocessed, and it shows a steady growth. It's also important that Metaspace is a community platform. So it is currently being used by more than 100 outlets, which represent a substantial portion of our field, and also many users. And over the just last couple of years, there were more than 30 publications of which were achieved where Metaspace was used, and many other review publications where it was mentioned. Here, let me present a bit more technical slide where I'm showing you how what is the infrastructure and architecture of the current serverful Metaspace. So it is running on AWS, on Amazon Cloud, where for processing for the custom bioinformatics, we spin out on-demand cluster used for Apache Spark computing, where we have Spark Manager and the worker nodes. And when someone uploads data to Metaspace web app, it triggers the processing on the cluster, puts the results, puts the data sets into storage, puts the results in database, and then the results can be accessed from the web app, but also from Jupyter notebooks with authentication, public results, or private results. And of the past, we have realized that serverless would be a perfect way of framework for generating, for developing a new version, the next level version of Metaspace. First of all, because currently we use fixed size cluster, and although, the data sets which are coming to us, they're coming from different labs, from different scenarios, from different type of experiments. So sometimes we underuse the capacity. Sometimes we actually it creates a bottleneck. And it's really hard to predict the job size just based on the data set, because it also depends on the molecular content. So we need to actually have flexibility in scaling up very dynamically. So second, it's actually turned out to be a substantial challenge to manage the cluster startup and shutdown. In particular, it's done in some custom way. So we have hundreds of lines of code to manage it and support it. Despite the fast network, which is used on AWS, sometimes there are bottlenecks created by Spark copying data between these worker nodes. And this in turn leads to the bottlenecks in processing, so some delays, but also sometimes increase of the cost. And also sometimes the cluster would fail, and then it stays running, so which leads to additional costs, prevents jobs, and leads to the requirements for additional maintenance. So in this respect, serverless would provide a perfect, scalable, but also easy to manage and ideally low effort architecture for Metaspace. And this is particularly important because our team is very small, and we try to really minimize the development costs and also maintenance costs. At the same time, providing services to hundreds of users across the globe. So we've been working with the Cloud Button project partners, and we have achieved, I believe, substantial progress of the post-half project of Cloud Button. And here I'm presenting you the midterm progress of Metabolomics use case in Cloud Button. So first and foremost, we have developed serverless architecture. And so what is important to understand for our use case is that we have every data set that is being uploaded by a user is screened for molecules from a database. So, and the processing step, it involves some steps on the data set, but also on the database. And then of course they emerge, and there is some screening and molecular search here performed. And here we are showing some estimates for the times necessary for individual steps. And we have also very carefully selected the steps which need to be benchmarked to compare serverful and serverless architecture. Next, together with IBM, we have developed a serverless version of the Metaspace computing engine. And if you're interested, you can go to our GitHub /Metaspace 2020. And there you'll find PyRen annotation pipeline, which is the serverless implementation of Metaspace annotation pipeline for IBM Cloud. And here you see the main contributors to this repository. Here you see that this work was uninterrupted with a substantial number of commits, more than 200 commits performed over the last 1.5 years, and two main contributors, Omer from IBM and Lachlan from Embo. So, and currently we have a version which can be used for benchmarking to compare it with the serverless. To prepare benchmarking, we worked together internally, but also with our collaborators, and first selected example data sets. And here you see the list of data sets which we made publicly available on our GitHub. And you see that they are provided from our collaborators, University of Rennes, or a company, pharma company AstraZeneca, University of Gießen, and also from us. And these data sets were selected because they cover a lot of different scenarios. Some of them they're small, some of them they're big, and some of them they have a lot of molecules, some of them they have not that many molecules. So they really help us cover the variety of almost all possible scenarios that we are performing in Metaspace. We also prepared and shared databases. Molecular database contain a variety of different molecules, because again, in every data set, we'll be searching molecules from one of these databases. And here we have we selected the default databases, which are used in Metaspace, but we have also specially prepared here a small database to test for small upcoming scenarios. And currently, we are not providing this in Metaspace, but this is one of the scenarios that we want to actually enable using serverless, as well as very big databases, which contain many more molecules that currently we are able to process. And this is, again, another scenario that we hope to enable using serverless architecture. So we have prepared the benchmarking data, also criteria for every benchmark, for every experiment. We have outlined what do we want to optimize, or what do we want to compare the serverless version against the serverful. Moreover, we have prepared Jupyter notebooks for these scenarios, and you can find them also on our GitHub repository, where we have outlined 3 experiments. So experiment 1 represents a typical use case. And this is more or less an average use case or average data set which is being processed on Metaspace. And here for performance, we would like to use definitely the processing time. But we also want to measure the latency time. And for every of these metrics, we also have outlined what kind of goal do we need to make sure that serverless version is at least as usable as serverful one. So peak memory usage, definitely cloud providing costs, but also developer time, because we expect that serverless version will provide us with the same functionality at the same time with a less development time and less maintenance time. In experiment 2, we hope that serverless architecture will enable us interactive reprocessing of the data. Currently, it is possible. However, it is not very user-friendly because one needs to spin out the cluster, install the Metaspace Apache Spark version there. And this takes a few minutes of delay. It's not that interactive. It's not that intuitive. And we hope that with the serverless, we achieve almost instantaneous setup and processing, but with the same processing time. And the experiment 3, we have prepared the Jupyter Notebook, but also a combination of data sets, benchmarking data sets and database to really perform a stress test of both serverless, upcoming serverless, and serverful implementation. So currently, we are using these Jupyter Notebooks together with our partner in Cloud Button IBM to really get comprehensive benchmarking results. They're not ready yet, but we're pretty close to this point, and we hope that we'll be able to achieve them in the beginning of the second term of the project. We have already achieved quite substantial development, but also we, despite the benchmarking results are not available, we believe that serverless is a very important technology for Metabolomics. And here we prepared, together with our partners from IBM Research and IBM blog post, which is called Decoding Dark Molecular Matter in Spatial Metabolomics with IBM Cloud Functions, where we discuss the importance of serverless in this field, where we discuss that for modern science, there is really the need of modern computing. And we outline how it was performed, and we outline that, yes, currently, in particular with the PyRAN IBM implementation, it is possible to move even such complex bioinformatics as Metaspace to serverless architecture. Summarizing the progress in the first term of the Cloud Button project. So I would like to list here the key achievements of EMBL, but also achieved together with our collaborators in Cloud Button, in particular IBM Research. So we have provided data sets and databases for benchmarking, but also outlined what are the experiments which we want to use for benchmarking, and what are the metrics and the goals for every metric that we want to achieve. We supported implementation and worked together with IBM Research to really implement it together for PyRAN IBM Cloud implementation. And we also integrated PyRAN implementation into the Metaspace pipeline and re-implemented parts of this pipeline for PyRAN IBM focused performance. We have provided code to IBM Research for testing and validating the logic of the custom bioinformatics in Metaspace workflow, and outlined the bottlenecks, in particular the bottlenecks in development, in the support, in maintenance, but also the upcoming cloud costs. And we started working together with IBM about, what would be the next evolution? And we're thinking that potentially a hybrid model would help actually address some of the potential bottlenecks which we're still waiting for the final benchmarking results, in particular for the cloud costs, but maybe also for development costs. But this, as you will see, this brings us to the next steps. So, and in the second term of the Cloud Button project, we expect very soon to receive final benchmarking results from IBM for our experiments, benchmarking experiments. We will start using PyRAN IBM Metaspace to apply to scientific questions in spatial metabolomics, which were not feasible earlier with our serverful implementation. We also plan to offer Pyro and IBM Metaspace implementation to scientists for beta testing, because there are several scientists in our team, but also externally, who use Metaspace using Jupyter notebooks, and they will be the first whom we actually offer to use the serverless implementation to achieve high interactivity with the results, but maybe even faster processing, and thus enabling new use cases. And we definitely are looking forward towards using this version in the production version of Metaspace. And finally, so if we see that indeed there are still some bottlenecks which are not addressed by serverless implementation of Metaspace, we'll continue discussion with IBM Research whether a hybrid cloud model combining both serverless and serverless would help us to address and achieve even higher improvements than just serverless alone. With this, I would like to wrap up the presentation. I would like to thank everyone who contributed to this work, in particular Lachlan Stewart from our team, and Gil Vernick from IBM Research, and also all other partners who contributed with their advice and consulting, Pedro, the coordinator of this project, and definitely the European Union Horizon 2020 funding for helping us performing this work. And thank you very much for your attention.